I'm Rochelle Crosbie Watson, and today we're going to talk about the basics of characterizing dystrophic phenotypes in MDX mice. So, we're going to cover a number of experimental approaches, including histology. We're going to look at membrane damage assays, molecular assays. We're going to look at some of the physical features and physiology, as well as genetic and chemical manipulations that are possible with MDX mice. Shown at the bottom is a picture of wild type and MDX mice. They're side by side. As I mentioned to you before, you're hard pressed upon a gross inspection of these mice to distinguish the MDX from the wild type mouse. But when we look at the skeletal muscle, we can see distinct histopathological signs that show the consequences of dystrophin loss in the MDX mouse. So when we're looking upon histological sections of the animals, it's very easy to tell them apart from one another. I want to remind you of the function of dystrophin. So this is a schematic diagram showing you that the dystrophin protein is located just underneath the sarcolemma membrane. It's associated with a complex of proteins called the dystrophin glycoprotein complex. Dystrophin interacts with beta-dystroglycan, shown in pink, and beta-dystroglycan interacts with alpha-dystroglycan, which in turn interacts with laminin. On the intracellular face of the membrane, dystrophin interacts with a host of proteins, including actin, and actin is then connected to the sarcomere, which is the contractile unit in skeletal muscle. In addition to interacting with the dystroglycans, the dystrophin glycoprotein complex is also comprised of sarcospan and the sarcoglycans. The sarcoglycans are single-pass transmembrane glycoproteins. On the intracellular face, again shown in yellow, are proteins called the syntrophins that interact with a protein in orange called dystrobrevin, and also another protein called NOS. We're going to hear much more about NOS later on in the quarter. So mutations in the dystrophin gene cause absence of dystrophin protein from the skeletal muscle membrane. The consequence of that is a loss of the entire dystrophin-associated glycoprotein complex. Laminin is not lost from the sarcolemma, but is not connected to the dystrophin glycoprotein complex. As a consequence, the muscle has lost that inside-outside connection, and that causes and renders the skeletal muscle to be very susceptible to contraction-induced injury, which means that there are microlesions in the sarcolemma membrane as a consequence of loss of dystrophin and the dystrophin-associated complex. When skeletal muscle experiences these microlesions, that causes an unregulated exchange of skeletal muscle proteins like creatine kinase. In other words, scientists can collect blood from MDX mice or from DMD patients and find the presence of muscle-specific proteins like creatine kinase in the blood serum because they have been released from these microlesions that occur on the sarcolemma membrane as a consequence of contraction-induced injury. There are a number of cellular and systemic consequences to this skeletal muscle membrane disruption. Muscle fibers experience extensive oxidative stress. There's mitochondrial dysfunction. There's also vascular constriction, and we'll learn about the effects of vascular constriction in the pathology of disease. There's activation of satellite cells, inflammation, and fibrosis, as well as the presence of fat as a consequence of the loss of skeletal muscle in Duchenne muscular dystrophy. So many of the histological findings that we'll look at when we're inspecting skeletal muscle of MDX mice is we will see that similar to DMD patients, there is an extreme fiber hypertrophy. So the skeletal muscle cross-sectional area of a DMD fiber can be quite large. There's also the presence of very small fibers, and we'll see that on an H&E stain in a moment. There's extensive degeneration and regeneration that occurs in MDX mice through the activation of satellite cells in an effort to repair the damaged muscle. There is fibrosis, necrosis, inflammation, and adiposity. The methods that are often used to reveal these histological symptoms include hematoxylin and eosin staining, Van Giesen staining, and oil red staining. We're going to take a look at those. So here is a low magnification H&E staining from a quadriceps, an entire quadriceps transverse section from a normal mouse. And you can see that there's a large section of healthy looking fibers in this particular section from a normal mouse. Contrast that with a section taken from a dystrophic MDX mouse. Shown in the boxed region are areas of extensive necrosis and inflammation. Now, if we take a closer look at a higher magnification, this is what we're going to see. First, let me tell you that in MDX mice from ages 1 to 3 weeks of age, the muscle histology looks pretty normal. And by that we mean that the individual fibers are of fairly uniform size, the nuclei are at the cell periphery, okay? And then at about 3 weeks of age, the muscle starts to experience the sarcolemma damage, the muscle fiber degeneration, activation of satellite cells, and regeneration. And in a skeletal muscle quadriceps from an MDX, as well as other muscle types, the skeletal muscles are at different stages of degeneration and regeneration. So we'll see the presence of many hypertrophic fibers, as well as the presence of many smaller, newly regenerating fibers. Newly regenerating fibers express developmental or embryonic myosin heavy chain, so we can use that as a marker to locate those newly regenerating fibers. Also note that in the dystrophic muscle, there's a wide variety of cell sizes. Some sizes are hypertrophic, some are quite small. The nuclei are also, in general, at the center of the cell, which is a marker of a fiber that has undergone regeneration. This cycle of degeneration and regeneration continues pretty much throughout the lifespan of the MDX mouse. This eventually leads to fibrosis and infiltration of immune cells. So let's take a look at this higher magnification quadriceps transverse skeletal muscle section. You can see that the fibers are closely packed together and that the nuclei are at the cell periphery. The arrows are pointing out to you a few of the nuclei. Contrast that with a similar section taken from a dystrophic MDX mouse. You can see that there are many hypertrophic fibers and also many fibers that are newly regenerating and smaller. Also note that the nuclei are at the center of the cell. When a skeletal muscle fiber regenerates, the nucleus moves toward the center of the cell, and that's an easy way for us to find the regenerated fibers. We can quantitate the extent of central nucleation simply by counting the percentage of fibers that have central nucleation in normal versus MDX skeletal muscle. And in this example, you can see that MDX skeletal muscle is very high in central nucleation, which is a marker of skeletal muscle regeneration. We can also find areas, particularly in the diaphragm muscle, and in the MDX mouse, the diaphragm muscle is one of the most severely affected muscles. So the fibrosis accumulates in the extrastitial spaces, and oftentimes the fibers, the muscle fibers themselves, look like islands that are surrounded by fibrotic tissue. And you can see that easily in the upper diagrams. There's also an accumulation of adiposity, which stains red with an oil red, so we can easily pick up those areas that have fat accumulation, which is another marker of dystrophic pathology in MDX mice. In addition to these histological observations, we can also use common laboratory techniques to investigate the molecular mechanisms of disease. And one of the questions that scientists like to ask is regarding the presence of proteins that are at the sarcolemma membrane. And doing these kinds of experiments is really very easy. Skeletal muscle sections are taken and placed on a microscope slide, and usually for these experiments, the transverse skeletal muscle thickness is around 5 or 10 microns. The sections are incubated with antibodies. Now, if you want to do a co-labeling experiment or you want to detect 2 proteins at the same time, you would elect to use antibodies that were created in different species. In this example that I'm giving you, the sections are incubated with laminin antibodies, which were generated in mice at the same time that they're incubated with antibodies to the embryonic myosin heavy chain that were generated in rabbits. Now, remember that the embryonic myosin heavy chain detects newly regenerating fibers, because as a fiber is undergoing nascent regeneration, it expresses the embryonic form of myosin heavy chain. The sections are then washed and incubated with anti-mouse secondary. In this example, the secondary is conjugated to Cy3, which fluoresces red, and the anti-rabbit secondary antibodies are coupled to FITC, which fluoresces green. This allows us to distinguish the laminin from the embryonic myosin heavy chain in the skeletal muscle cryosection. Then the sections are visualized under fluorescence microscopy using two separate channels. An example is shown here. So you can see in the wild-type muscle the gorgeous, beautiful laminin staining, which is showing you where the extracellular matrix is. Remember that the extracellular matrix is surrounding each individual skeletal muscle fiber. And in the MDX sample, you can see that there are these bright, small, green regenerating fibers. So we're able to find and mark those regenerating fibers by using this embryonic heavy chain antibody. We also see that laminin is denoting where each of those fibers is located in the MDX fiber. So while laminin is expressed in the MDX mouse, remember that the laminin is not connected to the skeletal muscle fiber because of the loss of the dystrophin glycoprotein complex. This is an example of an experiment. These are not co-labeling experiments. These are individually labeled experiments. And skeletal muscle from wild type and from MDX mice is stained with antibodies to dystrophin, beta-dystroglycan, and caveolin-3. So caveolin-3 is a protein that's present in the sarcolemma membrane, and it is not affected by the loss of dystrophin. So I hope you can see that in the wild-type mouse, the dystrophin and the beta-dystroglycan beautifully light up and stain the sarcolemma of each individual myofiber. The MDX mice have a mutation in the dystrophin gene that causes loss of dystrophin protein from the sarcolemma, and that's revealed by the lack of staining of dystrophin protein in skeletal muscle. Similarly, if you remember at the beginning of the lecture, we showed that beta-dystroglycan, which directly interacts with dystrophin, is lost in dystrophin-deficient MDX mice. And that is further revealed by the absence of beta-dystroglycan from the skeletal muscle sarcolemma in MDX mice. So, we have a review question for you. Below are 3 cross-sectional images of a wild-type and normal mouse quadriceps muscle. And this section has been stained with antibodies to dystrophin, beta-dystroglycan, and laminin. Now, how would these antibodies stain a quadriceps muscle from the MDX mouse? Choose one. Right. The correct answer is B. As we showed you, dystrophin and beta-dystroglycan are absent in dystrophin-deficient mice, but laminin is stained because the MDX mouse still expresses components of the extracellular matrix. So, how do we investigate sarcolemma membrane damage? Remember that in the first schematic that we saw, loss of dystrophin causes absence of the entire dystrophin-associated glycoprotein complex, and that causes a disconnect between laminin and actin that protects the sarcolemma membrane from contraction-induced injury. That causes these microlesions in the sarcolemma membrane that lead to an unregulated exchange of muscle proteins with proteins in the blood. And so scientists can take advantage of that muscle damage and quantitate it. And there are 2 basic experimental approaches to do that. One is the Evans blue dye assay. And the Evans blue dye assay allows us to identify damaged fibers, and it's really only used in animal models, whereas the serum creatine kinase assay reveals to us the overall systemic level of damage and is really used clinically as well as in animal models. So let's take a look at the Evans blue dye assay. So in this assay, mice are injected with an Evans blue dye, and in visual light, the dye is blue, and the chemical structure is provided for you. So the mice are injected through a variety of mechanisms, either intravenous or IP, and the ears turn blue, the nose turns blue, and the Evans blue dye binds to albumin in the blood, which is why the various peripheral regions of the mouse turn blue, and the dye accumulates into muscles that have a damaged sarcolemma membrane. The muscle is then harvested, and the accumulated dye is visualized after sectioning and placing those sections on slides and viewing under a fluorescence microscope. Now, even though the dye is blue, it actually fluoresces red, and you'll see that in a moment. So I know this is strange, but here are images of a mouse with the skin removed, and the mice have been perfused to remove the excess Evans blue dye, and I hope you can see that different muscles are picking up the Evans blue dye, and this is probably a function of the skeletal muscle use, which has damaged the skeletal muscle, allowing that Evans blue dye to accumulate in different fibers throughout the mouse's body. If we were to look at the sections under a microscope, we'll see something like this. These sections are stained with laminin using an antibody, and the Evans blue dye is fluorescent, so we don't really need to use any stain to visualize the Evans blue dye. So in wild-type mice, this represents an entire quadriceps section. You can see many skeletal muscle fibers. They're outlined because of the laminin staining. And we don't really see any red fluorescence in the wild-type sample. But in the MDX sample, you can see, again, lots of wonderful skeletal muscle fibers that are outlined in green due to the laminin staining, but we also see distinct fibers that are fluorescing red. Those fibers are damaged. The membranes themselves are damaged, which has allowed the accumulation of Evans blue dye into those skeletal muscle fibers. So, another assay that's used both in animal models and in the clinic is to take blood and quantitate the level of muscle-specific creatine kinase. And creatine kinase is a protein found in skeletal muscles. It's not ordinarily present in blood, and creatine kinase converts phosphocreatine into creatine, and in doing so also generates ATP. So scientists have devised a coupled reaction in which that ATP is converted to NADPH through the addition of exogenous enzymes, including hexokinase and glucose 6-phosphate dehydrogenase. And then that NADPH is quantitated by spectroscopy, shown here. So blood serum is collected, and then a coupled enzyme assay is used to evaluate the abundance of NADPH at a wavelength of 340 nanometers, which is the wavelength that NADPH emits at. And then from that, scientists can calculate the enzyme activity per volume of blood serum, and that is often expressed in units per liter. So an example of that assay is shown here. Plotted is the serum creatine kinase in units per liter for both wild type and MDX or DMD samples. And you can see that there is a huge abundance of muscle-specific creatine kinase found in the blood of dystrophin deficient mice and humans. And that's due to the extensive muscle damage that's accumulated as a result of contraction-induced injury. So what we want to emphasize about the creatine kinase assay is that it's really measuring systemic muscle damage. In other words, you're not able to tell which or how many fibers are actually damaged. That information can come from the Evans blue dye assay. So what we've learned in this module is how to take the MDX mouse and evaluate it both histologically for signs of Duchenne muscular dystrophy, molecularly for the various proteins that are involved in causing muscular dystrophy, and also at the sarcolemma membrane level, where we can really quantitate the damage that's done to the sarcolemma membrane as a consequence of the loss of dystrophin. These types of experiments are critical to moving the field forward as we test various treatments and therapies for Duchenne muscular dystrophy in a preclinical setting. So I hope you've enjoyed it, and I'll see you in the next one.